Hello everybody, welcome to the lecture of microbial cell cultures and its applications. Now the first question that comes is that why do we need microbial cell culture or why do we need biotechnology or biological origin product? A industry that has revolutionized how we live today in a society, from energy to our comfort to vacation to transports to many other things, is actually a petrochemical industry. The petrochemical industry is in everywhere that we see. It's just not only oil for fuel production, but it also produces base chemicals. It also produces polymers. So typical products are ethylene, propylene, phenol, acetic acid. These are base chemicals. Methanol, again base chemical. Benzene, ethyl benzene, uh, styrene, formaldehyde, ethylene glycol, and this kind of stuff, and there are many more as well. So basically in the petrochemical industry we take out everything that we could from the oil that we dig, and then basically whatever is left is even used as tar. So the only industry that actually can replace a petrochemical industry in its breadth as well as in the availability of the product is a biotechnology industry. The typical products that biotechnology industry produces so far is consumer products like bread, plastics, inks. In plastics, for example, polyglutamic acids, which is a natural polymer and can be used for production of plastids. PHB is another example, polyhydroxybutyrate. In pharmaceutical industry, biotechnology products are also produced, which is for which they are mainly known. They are antibiotics like penicillin, erythromycin, antibodies. These antibodies are useful for not only new generation treatment, but are also useful for diagnostics. Biosimilars, these are biological products which are not under patent anymore. Proteins, hormones like cortisol hormone that is needed for if your pituitary gland is not producing those hormones, then you need it for mood elevation and a lot of other activities. Vaccines, well, everybody is waiting for COVID-19 vaccine, and biology and biotechnology products are the way to produce it. So here the challenge is not only that we can produce a vaccine that is effective, but or we can have a vaccine that is effective, but a challenge is also to produce a vaccine in such a large quantity and able to transport it and give it to the patients in a very short span of time while maintaining its efficacy. So this is a challenge of a different level, that you would need 100 crore, 50 crore vaccines in a period of 1 year, and this is, or 6 months or even shorter, and this is a challenge that can only be solved by the biotechnology. And in any case, vaccine can only be produced by as it's a biological product, and it can only be produced by biotechnology. Then we need vitamins like vitamin B12 that is necessary for animals, and vitamin B12 and other vitamins that are also necessary for humans. Then we need therapeutics, interferons. Again, um, these are products that are necessary to treat diseases and are important for the patients who are suffering from those diseases. We can also use biotechnology to produce base chemicals like acetic acid, methanol, ethanol, lactic acid, glycerols, aspartic acid, xylitol, sorbitol, as well as higher chain carbons that can be used as a fuel. And this all products can are basically one way or the other can substitute the biotechnology, the petrochemical products. This is why we need biotechnology or biological origin product. So the question then comes, the next obvious question is that why we haven't done it so far. Well, there are several challenges in that. A, we cannot produce them in enough quantity, or let me take a step back and tell you this, that we cannot produce them at all. If we can produce them, then we can produce them in not so much quantity, and let us say if we can even produce them in such large quantity, maybe we cannot purify or achieve a good purification in a reasonable cost. So, all this factor leads to a fact that petrochemical industry is still ruling the roosters, and we need more development and more research in the biotechnology products. But before coming to the research, we need to understand the basics, and this is what this this lecture is about, to know a little bit more about the biotechnological products and how to produce them, and how to basically use microbiology, molecular biology, downstream processing, and all those engineering and biology branch to actually come out to a product. Okay. So, what we want? What we want is, we want our microorganisms to produce compounds or products that we need. Okay, this is that simple. There is nothing different in that. This is what we demand. So, what we want is essentially a microbial cell factory, like you have a microorganisms that is producing a product. Now, the question is that, do we want one microorganisms to produce or multiple microorganisms to produce? Now, this is a relatively simple question. So, if you have a microorganisms, so something like this, okay, this is our microorganism, okay, it will produce 10 products, okay, in 10 hours. Okay. Now, if you have 1 and 2 microorganisms, so what they will do is they will produce 20 products in 10 hours. So, essentially with the increase in the number of microorganisms, we can produce more products at the same time, right. In the bioreactors, this is what we actually need, and in the bioreactors the number of microorganisms that are present are in the range of 10 raised to power 6 per milliliter to 10 raised to power 9 per milliliter. Okay. And this is basically what we use or what we need to produce the product, and that is what the case is. All right. So, this is basically how we basically produce the product. Okay. Now, what do we need other than that? Okay. So, A, we definitely need multiple cells, which is fine. Now, what do we want to do? Again, it is essentially a catalytic reaction or a chemical reaction, okay, that you have A and this goes to B. A is your substrate, B is your product. All right. So, we have an A that goes to B as a product, and to convert A to B, we need microorganism. Okay. So, that means we need microorganism, and I will add a S here. So, it is microorganisms. So, we need multiple microorganisms, that is okay. What else do we need? What else do we need is, we need substrate, right. We need a starting point where microorganisms can take that and convert it to product B, and that substrate is called as raw material. Okay. So, raw material is used to produce a product through metabolism of the microorganism. Now, another important point is time taken to produce it, okay. And this is an important point. For example, the requirement of glutamic acid or polyglutamic acid or MSG, monosodium glutamate, which is a taste enhancer and especially is present in many of the food products or processed food that you eat. So, it really tastes very well, and the requirement of that is in millions of tons, in millions of tons per annum. Okay. So, you cannot have A converting to B, okay. Let us say it is converting 100%, which does not happen, but let us say it converts 100%. So, 1 mole of A converting to 1 mole of B, but the time it takes is, let us say, 1 month. And if your requirement is 100 moles, okay, with this system you can only produce 12 moles. So, 100 moles per annum is your requirement, you can only produce 12 moles, okay, 12 moles in a year or per annum. So, of course, you will say that why I can't have another 8 to 9 of such system to produce 100 moles, but that is the problem. If I have another system that actually produce not 12 moles per annum, but let us say another let us say 50 moles per annum, 50 moles per annum, that would mean that I would need only 2 of such systems. And what this will do, 2 such systems instead of using 8 to 9 such systems, you can think. The answer is basically it will reduce the cost, and that is one of the major driver in all the processes that we design or produce or learn. There are only 2 things. One is, if that product cannot be produced by any other method than what we have now, then you have no choice. The second is that the product that you produce has similar quality from the other method, but at a cheaper cost, and this will govern which technology would go forward. All right. So, another thing is that time taken to produce. If you can produce a product at much smaller time than what it takes, then essentially you reduce your cost, and that is a very favorable thing in a production process. Then what do we need for microbial culture techniques? Okay. As I said, just like in a chemical reaction, okay, so we will go back to our old system of A converting to B, where A is our substrate, B is our product. Okay. So, if you have I am sure you have studied catalytic reaction, or A going to B or A plus B going C. In these reactions there are several factors that plays. Okay. Similarly, in this reaction where I put MO, so this is your microorganisms which is converting substrate A to product B. Okay. And the condition that we need is pH, okay, temperature, okay, nutrients, okay, and substrate. So, pH, what is pH? pH is essentially the concentration of hydrogen ion, H plus ion in the water. Okay. Now, all the microorganisms cannot work at all the pH values. So, most of the microorganisms work at a neutral pH, which is pH 7, where basically the H plus ion concentration, okay, is 10 raised to power minus 7. Okay. So, this is one thing. Some of the microorganisms like the microorganisms that are used in the leaching, so essentially recovery of metals from the ores, many of those microorganisms are very happy when the pH is 1 and 2, so very acidic. So, H plus ion concentration is very high. There are many microorganisms which are very happy when the OH plus OH minus concentration or H plus ion concentration is very low. So, either OH minus concentration is very high. So, that would be pH of 10 raised to power minus pH of 9, 10, or the H plus ion concentration of 10 raised to power minus 11 or 12, okay, or even lower. But in the case of biological product, most of the time the pH is normal. So, which is very a positive point or a good point in favor of biological product production or biological product formation, uh, because in most of the chemical reaction the pH or the solvents that they use are not very environmental friendly. So, this is also one of the plus point of using bioproducts is we use environmental friendly environmental friendly procedure, and that is why we are looking at the biotechnology to essentially replace petrochemical industry because of the emissions and environmental challenges and the climate change that we are facing. All right. Now comes the another point, which is temperature, this thing. Okay. Temperature, in the case of temperature, uh, again most of the microorganisms like room temperature. So, somewhere between 25 to 35 degrees, 37 degrees, 40 degrees, 38 degrees, and that is about it. There are of course microorganisms that survives at 55 degrees, 60 degrees, but not normally not higher than that, and there are some microorganisms that survive at lower temperatures as well, but this is a normal temperature range and we want to control that temperature range. Okay. Then comes nutrients and substrate. So, substrate is typically called for a carbon source. Okay. So, and nutrients are other requirements that microorganisms have. So, it's you can consider it as nutrient as pulses that you eat or the source of protein, and substrate as carbohydrates, so chapati and rice that you eat. So, this is a difference between these two. All right. Then comes the next part, which is purification, because you already know that in the case of microorganisms they have produced a product, but there are many other things as well, and that basically leads that you need to purify those products as well. Okay. And there basically comes the bioseparation. So, now what is the case here is that you have microorganisms. Okay. So, this is your microorganism. You provide substrate to it. Okay. It produces product. Now, typically what can happen is that this substrate can be taken up by the microorganism. So, it goes inside the cell and the microorganisms eat this substrate. In the case of the product, you have two scenarios. You have microorganism, and I will write this as scenario 1 and this is scenario 2. So, you have a microorganism and we have and the product is formed. Okay. So, this is our product. Okay. This is product P1. Okay. Now, what we have is in scenario 2 we have the like this, we have microorganism. Okay. Okay. And the product that is formed is somewhere here. And this is P2 product. Okay. In scenario 1, what has happened is, so this is scenario 1. In scenario 1, what has happened, that the microorganism has consumed the substrate and the substrate is converted to product and the product remained inside the cell. So, this is a cell that is intact and it remains inside the cell. While in scenario 2, the microorganisms has consumed the product, consumed the substrate and the product is formed, but product is formed outside the cell. Okay. In both the cases, the separation is different. Okay. So, we will have a look. So, scenario 1, okay, and scenario 2. So, we have a microorganism. Okay. This is our, I will write a short form MO. Here we also have a microorganism. Okay. This is MO. Now, in the case of scenario 1, basically our product is this. So, this is our product P1 and in scenario 2 our product is this. So, these are two different products. Okay. This is P2, P2, P2 and this is P1, P1. And basically here there are multiple steps that you need to follow in both the cases for recovery of the product, and this we will discuss in the subsequent classes. Okay. Now, what happens in the scenario 1 is you have the microorganisms, you break the cell wall of the microorganism. Okay. And once you break the cell wall, the cell would look like, not like this, but would be like this. Okay. Maybe some part of the cell wall is still intact, but the product actually can come out. So, the product here, this can come out, product here which is P1, P1 would come out from here. Okay. Now, you can, after this you can basically separate, but of course, with the product not only the product will come out, what you will have other than product is, so you will have product for sure. So, your P1 in the liquid that you will collect, you will have nutrients that you added, okay, like basically nutrients, substrate, okay. Then you have cell wall breakage and plus products produced by cell, which are, okay, produced by cell, but not P1. Okay. Okay. In the scenario 2, basically since the product is already outside the cell, no cell lysis is required. Okay. So, cell lysis means breaking of cell and this is not needed, we just separate it. Okay. So, now what happens is that once the cells are separated and the product is separated, there are subsequent steps to extract the product and increase its purity. Okay. You can extract the product, and after extraction of the product there is more purity. You can increase the purity depending on the class of compound. So, for example, if it is a chemical, for some industry more than 90% is fine, the purity level is 90%. If for example, it is diagnostic, then maybe 99% is required, and if it is therapeutics which has to be injected into the body, you would want 99.99% or even more. In some cases, for example, in the case of semiconductor industry, if you are the purification depends on the kind of usage, and in case of semiconductor industry, which actually has nothing to do with biotechnology, but there are sometimes a requirement of the product, like metal is 7N or 9N, which means 99.5 times 9% pure or 99 plus 0.7 times 9% pure. So, the purity of the product will depend on its application that you need. All right. So, Now comes, so now what we have seen is that why biotechnology is important, why do you want to use it in the first place, then what are the things that needs to be, what are the main processes in the biotechnology, in the microbial product formation and its separation. So, the first is the formation itself and then the separation. In the product formation, you need to know microbial cultural techniques, okay, how typically a microbial culture takes place, okay. So, other than pH temperature that we have seen, it also needs nutrients, substrates, okay. And then there are different kind of equipments for microbial culture, then types of culture growth, how you can mathematically assess them, and this all we go step by step, all right. So, for in the case of nutrients of microbial culture, so what are the nutrients that microbes need? We will have a look at it, okay. So, the first is that there are 4 typical elements that are required, okay, and I am not saying elements in terms of periodic table elements, but elements in general, combination that is required, okay. So, we need carbon source, okay, we need nitrogen source, okay, we need trace elements, all right, and then we need growth factors, okay. So, carbon source, nitrogen source, trace elements and growth factors. In most cases we need, the amount of carbon source required is quite high, because the requirement is also quite high, because whole of the microorganism, their DNA, proteins and everything is basically the backbone is carbon backbone, right, they are organics. So, it has a carbon backbone. So, that is why they would need carbon source to basically produce this backbone and grow them and grow them, develop them and process them and so on and so forth, okay. So, what are the carbon sources, okay. So, there are multiple ways this is done. In the laboratory, one of the this nutrient, substrate, carbon source or all the sources that I have talked about, carbon source, nitrogen source, cofactors, okay, and so growth factors, which are also in some cases considered as cofactors, and trace elements. So, all these 4 are present in the media, okay, and this media can be of different type, it can be synthetic media, it can be semi synthetic media, okay. In the case of synthetic media, what is there is that every component is known precisely, all right. So, essentially what happens is that in the laboratory you add exact amount of glucose, exact amount of ammonia salt, exact amount of iron salt, exact amount of phosphate buffer, exact amount of again phosphate source, exact amount of any metal that is required like manganese, exact amount of that salt. So, that media is called as synthetic media, where every component is known precisely, okay, and this is would be the right way to go forward. However, in many cases there are in micrograms requirement of something, of some components, trace elements, and this becomes very challenging to add everything, okay. In those cases, and also it has been observed with practice that if you use semi synthetic media like nutrient broth or Tiptacas soya broth or brain heart infusion broth, and this actually many times works quite well and the microorganisms grows very well, okay, they are very happy over there. All of this semi synthetic media contained peptone, beef extract, yeast extract, casein digest. So, essentially like for example beef extract, how it is being extract. So, it is basically what they have is they have beef and um, uh, and basically either by soaking them in the water or boiling them and just keeping the liquid while removing the mass, that, and that liquid once concentrated is called as extract. Okay. Similarly, yeast extract is also produced, and similarly in case of peptone, basically there are enzymes that digest the animal tissues, and that digested is called as peptone. So, basically they are we do not really know the exact composition of them. So, we cannot even say that it is composed of this compounds, and we cannot even say that it has this much amount of carbon, this much amount of nitrogen, this much amount of so maybe we can say something about carbon and nitrogen, but not really for all the other elements. So, that is why it is called as semi synthetic media, and we really do not really know exact composition, which so, but they are very good for the laboratory practices. Okay. You can just mix them in a known, so you can just take solids like let us say 10-20 grams of that in the known amount of water, and you are good to grow your microorganisms. The challenge come is that this all of the above nutrients are extremely expensive, and it is not sustainable for the large scale bioproduct. So, if you want to run a 1000-liter fermenter, which is not uncommon, the amount of semi synthetic media that you would need would be so high that it will not really go very well with you. Okay. So, your costs would be very high, and thus this would not really work. All right. So, what are the alternatives? Alternatives are cereals and other molasses and other products. Okay. But before selecting the alternative, you need to consider something else. You need to consider few points before selecting the media. So, the first point is that the media should yield maximum product or biomass per gram of the substrate used. What is yield is essentially the amount of biomass produced or amount of product produced per gram of the substrate, and this should be the maximum. That means that essentially if you are putting 1 mole of it, and then it should at least be converted 80%, 90%. Well, ideally we would like all of that should be converted to biomass or the product, but many times it does not really happen, but still the maximum it is, the better it is. You know why? The reason is that if we do it, essentially you would not require that much amount of biomass or substrate actually to produce a known amount of biomass. So, your yield is high. So, essentially we will go back to our old model, A goes to B, this is your microorganisms, okay. This is your substrate, this is your product or biomass. So, your product can be biomass as well as antibiotics, it can be antibodies. Okay. So, product can be anything, biomass itself is a product. For example, in the case of algae, biomass itself is a product. All right. So, what we want? What we want is that if 1 mole of substrate is consumed, okay, what we want is that let us say it produce 0.8 mole of biomass. This is your scenario 1. In scenario 2, it produces 0.82 mole of biomass. Okay. So, this is your scenario, this is your scenario 2. So, what the first statement that I read was, which was should yield maximum product or biomass per gram of substrate used. Okay. In that case, basically this is 0.80, this is 0.82. So, this one is better. Okay. So, that is what that statement means. Okay. Now, what is the second point? The second point is that it should be of consistent quality and available throughout the year. Now, this is very important, because if your quality changes, okay, then basically your whole of the bioprocess changes. Okay. The yield would change, the end product might change, definitely the contamination in the end product would change. So, essentially it changes whole of the process that you have designed. Okay. So, every process of course have some tolerance. So, if it is not as if that if 0.01% changes then it is bad, that will not happen. There is some amount of tolerance, maybe 2%, 3%, but any more deviation from that tolerance limit would lead to changes in your bioproduct. It would lead to changes in your definitely the contamination in the product, and then subsequently it will affect your bioseparation process. All right. So, essentially we need it to be of consistent quality and available throughout the year, of course, because you cannot depend on it seasonal, that okay it is produced now, so you can use it, that will not work for you. You need it to be available throughout the year, so that the process and the reactors can run throughout the year. Okay. Now, another thing is that it should cause minimum problems during preparation and sterilization. This point is important because it makes the process easier. Okay. But I will come back to this point when we are talking about preparation and sterilization in more detail. All right. Then it should give minimum problems during production process, particularly aeration, agitation, extraction and purification of the product. Okay. So, essentially this all leads to cost, and that is why we want minimum problems. So, it is not as if that we cannot handle the problem is we can handle the problem, but it takes time, it takes manpower and it takes additional equipments. So, all these three things leads to essentially increase in the cost of your bioproduct. Okay. Now, so in terms of problem, what I was saying was that we will go back to our old system, which is A going to B. Okay. A is substrate, B is our product, all right, and this is our microorganisms. All right. Now, we have substrate A, let us say we make this as A. In this case we have substrate A1, okay, and we have substrate A2. Substrate A1 is very good, okay, it gives product B plus some contamination, okay, contamination which is contamination 1, we can write it like this. All right. A2 gives again B plus some contamination 2. Okay. Now, let us say the conversion here, if we take 1 mole of this, 1 mole of this, the conversion here is 0.8 and conversion here is 0.83. Okay. From our previous discussion, we would agree that this is better, right. So, this is better, but the contamination 1 is you can remove this one by 99%. Okay. So, we can remove contamination 1 very easily by 99%, while contamination 2 actually we can only remove 80% easily. For next 19% we need to invest 1.5 times more effort. Okay. So, what happens here? Even when the 0.83 is better in the downstreaming or the contamination process, okay, we can remove 99% in this case quite easily, but here 80% can be removed quite easily, but for rest 19% we need 1.5 times more effort. So, basically that would mean that even if 1 mole converts to 0.83 and this is better, eventually this whole process would become very expensive and very hard to operate. Okay. So, this is just one example of contamination. Now, here you can even add that let us say A1. So, You can even add like this: A1 goes to B plus C1 contaminant 1, A2 goes to B, and again I will say contaminant 1 is the same contaminant. So, here that is not a problem of contaminant, okay. But the problem is this, that let us say this gives 0.80, this gives 0.83, okay, with 1 mole and with 1 mole, all right. So, this gives 1 mole 0.80, 1 mole 0.83, okay. Now, to convert 1 mole to 0.83, your agitation speed here is only 100 rpm. So, that is revolution per minute, but here because of the substrate there is problem of the mass transfer and things like this, you need to keep the agitation speed as 130 rpm. Okay. So, there is one thing that we actually talked about in this was mass transfer. So, this has been explained in detail in the subsequent lecture. You can simply take an example of perfume. So, a perfume, when this spread or put in the air in the room, what happens is that it will be only in one corner of the room, but if a fan is moving, then basically that perfume smell would spread across the room. So, essentially that is what mass transfer is, that you are putting energy like of fan and creating the mass transfer. Essentially it is just a movement of anything from one phase to another or even in a single phase, but it has major issues and major energy consumption points when it comes to bioprocess or chemical industry processes and so on. Okay. Now, even if 1 mole gives 0.83 and 1 mole gives 0.82, it is not straightforward that this system. So, scenario 1 and scenario 2. So, even if 1 mole gives 0.83, it does not mean that scenario 2 is good, because you have 30 rpm more, and this means you need to put more energy here. Okay. So, that is why it is important to look at the process holistically and through complete cycle, that how basically what will be the best choices. Okay. So, in the case of nutrient, we have discussed that so in the case of nutrient we have discussed synthetic, where you know each component, and semi synthetic like yeast extract, peptone, uh, um, tryptocase, soya broth and things like this. Okay. But these are only for laboratory scale. For larger plants or fermentations, basically the kind of carbon source we look at is our cereal grains, starch, cane, molasses, glucose, sucrose, lactose, okay. And these are the ones that are produced in large quantity. These are all agriculture products, right. Not glucose, sucrose and lactose, not purified ones, but cereal grains, starch, cane, molasses, these are all sugar, and then basically can be fermented to a product. Okay. So, that basically decreases the cost. You can buy a lot of cereals like wheat or molasses at a very cheap price compared to your soya broth or your peptone, all right. In the case of nitrogen sources, uh, again in the case of synthetic and semi synthetic media, we used to use peptone, and this were the again source of nitrogen as well. But here we use ammonium salts, urea, corn steep liquor, slaughterhouse waste, and basically these are a very good source of nitrogen and extremely cheap. Okay. And all of this nitrogen can be utilized by microorganisms. So, that makes them very good to use, all right. Then the third is trace elements. Uh, Trace elements such as iron, copper, manganese, molybdenum. So, these trace elements are required because we need these elements for effective working of enzymes. Okay. The enzymes can only work when these trace elements are available. If these trace elements are not available, then the enzymes cannot work, and they require them in very minute quantity, in microgram per liter, but they would nevertheless need it. If these trace elements are not there, then essentially your reactor would not perform to an optimal level first, and then eventually this reactor would die. Okay. So, you really need trace elements. Okay. A very typical example of is anaerobic digester. So, this is not a fermentation bioreactor, but anaerobic digester, that means basically under anaerobic condition you are producing biogas, and in that cases many times you need a cocktail of trace elements to for the anaerobic reactors to work effectively. All right. Then you also need growth factors like vitamins, amino acids, because in many cases these vitamins and amino acids are not produced by the microorganisms. So, they need it to be supplemented. In peptone and this kind of semi-synthetic media, you already have vitamins and amino acids present in the by natural source in this media, but in our case for nutrients we really need to add them separately, but again they are required in a very small quantity. Then you need energy sources. So, basically you need microorganisms to use energy. So, they would use energy from this nutrients that you give to convert them to convert the substrate to product. All right. And in the meantime they also gain energy through it. Okay. So, for example, in carbohydrates the carbon is reduced, and basically if you oxidize that carbon it forms carbon dioxide, and this carbon in the meantime this will also give the energy to the microorganisms. So, this is kind of a respiration process. Okay. So, that is not necessarily to happen in all the cases, but that is how the energy source and things like would work. Okay. What are the other requirements for the microbial culture? Okay. One of the requirement is antifoaming. Okay. Now, how does the foam are produced, and why does the foam is a challenge? Okay. But in principally what the foam are actually stable gas bubbles, okay, or there are small pockets of air in liquid. So, these are, or you can say as I said earlier, they are stable gas bubbles which does not burst very easily, and the presence of foam has a major challenge because it interferes with the sensors and the probes that you would have in the bioprocess. So, for example, if you have an oxygen sensor in the bioprocess, or you have a pH sensor in the bioprocess, because you want to maintain those two parameters essentially to control your bioprocess, and because of the production of foam it may interfere with it. Also, another challenge that comes with the foam is that it actually takes up the liquid also. So, if you are removing the foam and if there is a product that is in the foam, you lose it. Okay. So, foam is produced whenever you take a bath and use soaps, basically foam is produced. Okay. So, soap is actually a foaming agent, not an anti-foaming agent. Okay. Why soap is a foaming agent? So, basically soaps are or in normal terms your surfactant is a foaming agent. And what is a surfactant? Okay. And soaps are also kind of a surfactant. Okay. They are amphiphilic molecules. So, they have hydrophilic parts, or okay, and they also have hydrophobic parts. Okay. And because of this they reduces surface tension and allow the formation of foam. Okay. But what in principally foam is? Foam is small pockets of air in a liquid. Okay. So, in a liquid, in a continuous flow of liquid you have air bubbles. Okay. And this air bubbles are actually foam. In the case of surfactant or soaps, basically the quantity of air bubbles is very high because they reduces the surface tension and promotes the formation of bubbles, also prevents the bubbles to become too big and then collapse. Okay. So, because of that surfactant becomes are foaming agents and they lead to production of foam. But how does it matter in our case in the bioreactors, right? Means we are not adding any soap in the bioreactors. So, why we are having foam in the bioreactors? Well, the answer is, can think of it. I give you 1 second, 30 seconds to think about it. So, the answer is proteins. All right. Proteins have hydrophilic parts as well as hydrophobic part. You know that proteins are made of 20 amino acids. In some of the amino acids like alanine is hydrophobic, while aspartic acid or glutamic acid are hydrophilic amino acids, right. In some cases they have these amino acids then combine together, form a peptide bond and become a big long chain of protein, right. So, because of this the protein becomes quite big, because of this amino acid combining each other and making peptide bond, and of course, forming proteins which are very essential for the growth and everything that microorganisms does, okay, or do microorganisms do. It is very essential. So, proteins are there and this protein actually give the forming properties. Okay. So, what is the challenge? We now know that what is a form, how forms are formed through surfactant. In the case of bioreactors, the surfactant is proteins. Okay. This protein leads to the, or there can be other compounds as well, but mainly proteins. This protein leads to the forming, but what is the challenge of the forming? The challenge of the forming is that in many times you have a gas liquid interfaces, your liquid also get trapped in your air bubbles, okay, and it starts to go up. So, basically if your reactor, if it is 800 milliliter or 1 liter reactor, you will have 500 milliliter of the water that you could see and the rest is foam and all the rest of the liquid is trapped there. Now, what would happen is that this foam would not settle down and essentially many of your product would end up in the foam, okay, and then basically you are losing the product in the foam. So, essentially it can be something like this, that in normal case this is your bioreactor, okay, and this is the level of water here, okay. In the case of foaming, so which would be something like this, okay, your water level is only till here, okay, that you could see, but your forms are here. All right. So, basically this much amount of volume of water, okay, this volume is here trapped in air bubbles. Okay. In this air bubble only not, so if the volume is trapped, that means your product is also trapped. Okay. So, you do not really want that. So, what do we do? What do we do is we basically break the foam. Okay. So, we again increase the surface tension and break the foam. How do we do it? You can add olive oils, you can add sunflower oil, you can add silicones, that increases the surface tension, and that would lead to breaking of the foam and your product that is trapped in the air bubbles would come back. All right. Now, one major requirement of a microbial culture and microbial growth is water. Okay. So, microorganism. So, in the case of chemical reaction, chemical you can use different kinds of solvents, but in the case of microbial products or bioproducts, the media or everything is done in water. Okay. The liquid phase is water. Okay. So, essentially you need a large quantity of water, okay, but it is not as if that not that large quantity as you need in agriculture, but you need quantity of water, enough quantity of water. But more importantly, you need water that is clean. Okay, you need um, water which is more clean than your drinking water in many cases. Okay. And you also need to make sure that in your water there is no other microorganisms or it's not contaminated. Okay. You need to know that your water does not have any other mineral salts that can affect the growth of the microorganism. That's why you need a clean water. Okay. Um, for example, typically in your drinking water you can have 50 to 55 milligram per liter of sulfate, while such a sulfate in a media, especially if you are doing especially if you are running anaerobic, then this sulfate can actually reduce to produce sulfide. So, the point is that you need a water that is clean. Okay. You also need in the water to control the pH. Okay. So, you cannot, it may happen in a biological process that your pH is not controlled, and that is why you need buffer. And why it can happen? It can happen because of a simple reason, that we go back to our original equation, A goes to B. Okay. So, this is your substrate, this is your product, but you can also produce M here. Okay. So, M, you can also produce C here. So, C is contaminant. Okay. So, contaminant can be something which is, well, M is also typically is M is also a contaminant, but I am just differentiating it over here. M is your metabolite, okay, or something, any chemical something that actually changes the pH. So, changes pH. It can be acidic, it can be intermediate, it can be anything that changes the pH. And if basically it changes the pH, then essentially you need pH control. Okay. So, pH control can be done by buffers, where you already add enough that any kind of change in the metabolite is not leading you to change in your conditions. Or the other option is that you have a dynamic control, where essentially what it means is that you have a bioreactor and you have pH sensors. They are measuring the pH continuously, and as and when pH moves in the direction, they add. So, if the pH moves in acidic direction, they add a base, and if it moves in the basic direction, then they add an acid to just maintain the pH. Okay. So, this is also very important. Third thing is that we also need water for ancillary services like heating. So, in case your room temperature goes 10 degrees or 18 degrees, you need to heat it your reactor to 25-30 degrees, whatever temperature you are maintaining. So, that is one. Second is that you need water for autoclaving or sterilization, which is a topic which we will study after this. Okay. So, you need for that as well. Then you need the water for cooling in case the temperature has gone high. Okay. So, you need water for cleaning, you need water for rinsing. So, you also need a good enough sufficient supply of water. Without water you cannot really perform the produce a product. Okay. So, the next is cultural procedures. Okay. And in cultural procedures we study the sterilization procedures and aeration and mixing. Okay. With that we will end this lecture. Thank you very much.